mainly divided into the background, the introduction to basic concepts, and application and prospect in disease treatment. So without further ado, let's get right into it. To begin with, I will introduce to you the background of stem cell therapy technology and disease treatment. Since the 1990s, American scientists have successfully grown human embryos. After stem cells, the research of stem cell-directed differentiation and its regulation is the hotspot of life science now and in the future. Developmental biology and its related disciplines will play a breakthrough role in promotion. Meanwhile, this field has great medical and commercial value, as well as scientific significance. It is bound to be the key support and development of this field. In 2003, significant advances were made in basic stem cell research, gestational stem cell research, and adult stem cell medical research, which not only gave hope for the fight against disease and stimulated medical research, but also intensified the debate over different ideas and policies. The Bioethics Steering Committee of the United States studies the scientific, ethical, and legislative implications of human embryos and provides extensive and in-depth commentary and guidelines for human embryonic stem cell research. Stem cell technology has a very attractive prospect not only in the treatment of diseases, but also in the cloning of animals, transgenic animal production, developmental biology, new drug development and efficacy, toxicity evaluation, and other fields will have a very important impact. Stem cell technology as a cutting-edge technology in biotechnology has become the most eye-catching field in natural science and is considered by the medical community as a milestone in human life science research, which indicates that life science research will enter a period of rapid development and is an important basis for the renewal of high tech and the development of emerging industries in the future. All right, next I will explain to you some basic concepts of stem cells and stem cell technology. Stem cells are self-renewing pluripotent cells, keep the state of undetermined differentiation and proliferation ability under the right conditions or give the right signal. It can be differentiated into a variety of functional cells or tissues and organs. The medical circle called it versatile cell, also known as the cell that can do everything. Stem cells come from embryos, fetal tissue, and adult tissue. Stem cells are divided into embryonic stem cells and adult stem cells according to their developmental stages. The differentiation and proliferation of embryonic stem cells constitute the basis of animal development, while the further differentiation of adult stem cells is the basis of tissue and organ repair and regeneration in adult animals. Stem cells are divided into topotent, pluripotent, and unipotent stem cells according to their potential to differentiate. Undergo programmed death or apoptosis, hematopoietic stem cells can be enriched by their physical properties and cell surface characteristics. Hematopoietic stem cells are derived from bone marrow, peripheral blood, and umbilical cord blood, which can differentiate into various cell types of blood and immune system. They can be used for the treatment of leukemia, lymphoma, and hereditary blood diseases, such as various hereditary anemia, congenital metabolic disorders, and the recovery of hematopoietic stem cells after treatment of cancer. Many studies have found that bone marrow, mesenchymal stem cells, have been isolated from the bone marrow of humans, birds, and rodents. These stem cells share the common characteristics of stem cells. More recently, mesenchymal stem cells have been isolated from human skeletal muscle. Therefore, using mesenchymal stem cells for tissue engineering research has the following characteristics. It is convenient and harmless to the body. Mesenchymal stem cells can be obtained by a simple bone marrow puncture. There are no problems of tissue matching and immune rejection when transplanted from autologous tissue. Mesenchymal stem cells can be differentiated into a wide range of tissue types and can theoretically be differentiated into all mesenchymal tissue types: bone, cartilage, muscle, tendon, which has the application value in the treatment of traumatic diseases. Dividing it into cardiac tissue would make it possible to build an artificial heart. It can be differentiated into dermal tissue and its main function is to act as a backup reserve involved in the repair of nervous system injury and regeneration of normal cell death. Last but not least, I will introduce the application and prospect in disease treatment. Gene therapy of hematopoietic stem cells. Many diseases, especially some hereditary diseases, are associated with hematopoietic stem. With the development of hematopoietic stem cell transplantation and gene technology, a new therapeutic method that tries to fuse the two technologies, gene therapy of hematopoietic stem cells, has become a hotspot of current research. Many diseases, especially some single gene diseases such as hemoglobin disease, sickle erythrocyte anemia, thalassemia, chronic granulomatous disease, and immune deficiency disease, are related to the inherent defects of hematopoietic stem cells. In principle, these diseases can be treated by gene replacement. The basic idea of hematopoietic stem cell gene therapy is to mobilize the hematopoietic stem cells isolated from patients to transfect target genes in vitro, and then do autologous stem cell transplantation to replace congenital diseases caused by gene loss or damage in hematopoietic stem cells. In anti-tumor chemotherapy, drug-resistant genes are introduced into normal hematopoietic stem cells to produce chemotherapy-resistant proteins, with ultimate goal of reducing or canceling the toxic and side effects of chemotherapy. As an intercellular vaccine, such as transfection of antiviral genes into hematopoietic stem cells or T-cell cursors to resist HIV infection. Of hematopoietic stem cells. A variety of cell surface markers have been used to identify hematopoietic stem cells. The cell surface model of hematopoietic stem cells in bone marrow is CD34. Baboons were irradiated with lethal dose and transfused back CD34 plus enriched bone marrow cells can reconstruct the hematopoietic system of baboons, indicating that CD34 plus cells have long-term multilinear differentiation ability. At present, there are very mature methods to obtain CD34 plus cells in clinic. Theoretically, isolated and concentrated CD34 plus cells can improve use factor usage. Selection of carrier. A variety of transfection systems have been used for hematopoietic system cells, such as DNA, liposomes, adenovirus vectors, adeno-associated virus vectors, adenoretrovirus vectors, and lentivirus vectors. Optimization of transfection conditions. Transfection efficiency is the bottleneck of gene therapy. In order to improve transfection efficiency, a series of attempts have been made on the external conditions of transfection. In this study, co-incubation with bone marrow stromal cells was found to improve the transfection efficiency of human stem cells transfected with reverse rate viral vectors, suggesting that exogenous stromal subfractions influence gene transfection. Fibronectin is a well-studied molecule that connects both vector particles and stem cells, further providing growth promoters to improve transfection efficiency. The incorporation of cytokine polypeptide sequences into envelope protein molecules, such as the incorporation of I2 into chimetric envelope protein, is highly efficient for transfection of IL-2 dependent cells. Limit the efficiency of hematopoietic stem cell gene transfection. Although the efficiency of gene transfection has improved greatly, gene therapy of hematopoietic stem cells has not been very successful. The main factors are listed below. So number one, the original hematopoietic stem cells were in a resting state, and surface receptor level was very low. At present, there is no very effective vector, and the efficiency of transfection is very low. After successful transfection, the target gene often cannot be expressed effectively or for a long time. Lack of effective means to select and transfect successful hematopoietic stem cells. The original hematopoietic stem cells were in a. All right, I guess this page was the same as the last one. I didn't change that. Sorry. So I'll just repeat this part. So number one, the original hematopoietic stem cells were in a resting state, and the surface receptor level was very low. And number two, at present, there is no very effective vector, and the efficiency of transfection is very low. Number three, after successful transfection, the target gene often cannot be expressed effectively or for a long time. And number four, lack of effective means to select and transfect successful hematopoietic stem cells. Hematopoietic stem cell transplantation for the treatment of autoimmune diseases. Hematopoietic stem cell transplantation, which is abbreviated as HSCT, for more than 10 years. The treatment of refractory autoimmune diseases, which is abbreviated as ADS, has achieved gratifying results. Domestic and foreign scholars have classified ADS with rapid disease progression resulting in obvious disability or death as severe autoimmune disease, which is abbreviated as SADS, and its expected life period is 5 to 10 years. With the development of science and technology in recent years, the treatment-related mortality rate of autologous hematopoietic stem cell transplantation is 1% to 5%. Aggressive treatment is justified and necessary. Hematopoietic stem cell gene therapy reflects a new treatment idea, although the basic research stage brims with the development of hematopoietic stem cell transplantation and gene technology. Besides, with the knowledge of the genes themselves, hematopoietic stem cell gene therapy will be fundamental to cure a batch of diseases. All right, and that's all for my presentation. Thank you so much for listening. Thank you.
Thank you so much, Yuseng. So I open now the floor for some questions or clarification for Yuseng. Anyone? Would you like to ask Yuseng about his report? It was well presented, Yuseng. Congratulations.
Doc,
Yes, go ahead, Peter. Is that you? Go ahead.
on cam daw, Doc, sabi ni Sir Jem. Joke lang.
Okay. Yeah, all right. Okay, Alexander, do you have some insights or inputs with regards to this topic? This is a very interesting and timely, technology, cell therapy using stem cells. So you're saying I have go ahead. Sige, go.
No insight po, Doc.
Okay, all right. You're saying I just have a quick question. So currently, we are not allowed, no? As in, researchers, scientists alike are not allowed to do embryonic stem cells. Is that correct? Only adults.
Yeah, that's right.
Only adult stem cell. Yeah, not embryonic. No? Because there are many ethical issues surrounding the use of embryonic stem cells.
Yeah, and also some like ethical issues, right?
Yes, that's correct. Okay. All right. So, any other insights? Would you like to add some more? I have prepared a few slides just to add some more insights with regards to stem cells. So, maybe I can share one of my slides, and then in case you have some inputs, then please go ahead and share with us. All right. So let me just put that slide. Oh, wait a minute. So, all right, here. So this was already mentioned earlier, no, by our reporter. So yeah, one of the so-called standard of care treatment, no, as far as leukemia patients are concerned, is bone marrow transplant, and this is already well established and already standardized, no. So as you can see in this figure, no, the first step is to be able to isolate, no, to collect the stem cells from a prospective donor, all right? And then from this, we start to culture, to propagate it, no, in vitro, all right? So we put in the necessary ingredients, no, like the media to support the growth of these stem cells so that it will later propagate. And later on, we do the priming, no, we prime the cells, and once primed, they are now transplanted onto the recipient. So mostly, those with cancer, like leukemia patients, they are the best candidates to receive this standard of care treatment, like the bone marrow transplant. So there are a lot of hospitals doing this right now, and the success rate is quite impressive. Nonetheless, those who undergo bone marrow transplant need to be monitored. No, so there are a lot of assays that are requested, no, from time to time to monitor the success of this transplantation. Now, you might ask, aside from bone marrow transplant, what are other so-called standard of care treatment being done, let's say, in the country for that matter? Well, I'd like to to share this with you. This was our experience in the medical center in St. Luke's. Okay, so this one is about the so-called ocular stem cell transplantation. All right. Back then, it is not part of the standard of care treatment, but because of a series of studies and several attempts already, we were given the go signal, no, to go ahead and offer this as a service to our patients who would need not a bone marrow transplant, but in this case, an ocular stem cell transplantation. So basically, the steps are quite similar to the previous one that I presented. But in this case, you have a patient, let's say, suffering from a chemical injury. Like in this case, I think this is the left eye of a patient. No, I won't be giving the details about this, but because of some quarrel, no, the suspect, no, spilled no chemical to the patient's eye, and so that causes this injury. So to make the long story short, the unaffected eye, I think the left eye, which is unaffected, we collected no stem cells from that eye, no, through the help, of course, of our physicians, no, our specialists, and we cultivated it in vitro in the lab, no, yeah, and so it took several days to cultivate that, and prime the cells, and then later on, these cells, no, that were primed already, were transplanted back to the patient to help restore the vision. And the outcome, the final outcome was the patient recovered and was able to see then after several weeks. So this is one success story, no, of stem cell therapy, because as mentioned earlier by the reporter, with stem cells we can prime them and we can program them such that they can develop into a mature cell that can that can perform its function, so to speak. So again, aside from your bone marrow transplant, aside from your ocular stem cell transplant, these are some of what I came across, no, as far as your stem cell based products. So there are a lot of products available now in the market, no, and the different kinds of stem cells. So from human hematopoietic stem cells to mesenchymal stem cells, no. So these are some of the published stem cell based products and their corresponding indication, from osteoarthritis to Crohn's disease, from graft versus host disease, no, even you have a bone and cartilage degeneration, and heart infarction, to name a few. And as to the types of transplantation, so you have this term allo, meaning from another donor, or auto, no, from the same patient, just like in the ocular stem cell. So the patient was the donor of that stem cells, no, from the unaffected eye. It was transplanted dun sa affected eye, no. Yeah. So there are several countries, no, producing this, right? But I'd like to caution everyone, no, that well, this is quite interesting, but at the same time challenging, because you really have to have the permit to do so, no. So I'd like to share that one, no. So let's say the institution would like to go ahead and offer this as a service. One of the important things that the stakeholders will do is, number one, that they have to have a GCP certificate, no, good clinical practice. Here, the focus is on the rights, the safety, and the well-being of the participants, no. So that's number one. Number two, in the laboratory, because this is where we will cultivate, we will propagate the cells. So the laboratory should demonstrate a high standards, no, quality standards in the conduct of these studies, no? And then, of course, you have the GMP, no? This is good manufacturing practices, because we already have this service, no? So we have to sustain, no, these manufacturing practices, no, being mindful of the quality control, no, because even a small contaminant, no, a virus, for example, can contaminate the entire batch. No, so we have to be very careful in doing this, and so that will be very crucial in making stem cell available to patients. All right. So that will be my insights for this topic. Okay. So going back, would you have? Yeah, I heard somebody.
Doc,
Okay. Yes.
just a question, Doc, about the
Yes. Yes.
service on the ocular transplant.
Yeah. Yeah. Oo.
How much is it, Doc? Is it free or
Yeah.
yeah,
Medyo
I
I
know
lost. Oo. So it's actually ano Peter, no? It's a package. There's the use of the facility. That's one. There's the PF of the attending doctor. I think mga close to the last one I heard is about mga 200,000. All right. So
okay.
more or less. Kasi
Okay.
it's very ano oo. So yun yung mga challenges doon. But it's a promising ano naman, it's a promising treatment no to those patients that would need one. No? Kasi nga this is a serious ano problem like yung chemical eye injury. So the way to restore it is stem cells.
Doc, can it be used as a replacement for LASIK as well? To those with problem with what do you call those? Yung farsightedness, Doc, and short-sighted. I forgot the...
Oo, yung mga yeah, oo. That I'm not very sure, no. Probably, yeah, with the use of stem cells, no. Pwede rin makorek yung kanyang vision, yung kanyang visual acuity, no, ma-improve. Yeah.
And then, Doc, I think Sir Jem has a question for the reporter.
Yes, go ahead.
Sir Jem, yeah, your question.
Go ahead, Jem.
Doc, actually, my question is for you.
Yes, oo.
Doc, regarding the procedure, is it 100% effective among all the patients that you had?
Sorry, medyo choppy. Go ahead, Joe.
Regarding, say, the procedure, is it 100% effective amongst all the patients that you had that had undergone the procedure po?
Yeah, oo. Yung clinical outcome was quite what do you call this? Encouraging. I don't have the exact data, it's with the clinicians actually, but based on the number of patients who underwent this kind of treatment, we had more than 10 na, no? More than 10. That is why it is offered na as a service to those who in need, no, of these kinds of therapy. Go ahead, Alex.
Hello? Doc, this is regarding the bone marrow transplant po.
Yes, bone marrow naman.
Is it for like all types of leukemia? And is there sort of like a panel? Like if this treatment is going to work or something like that?
Mm-hmm. Yes, so but basically itong bone marrow transplantation is ordered by the oncologist with consultation with a hematologist as well. So they team, meron silang team, so they request for this form of therapy, no? And may mga ibang mga test pa na ginagawa before and even after treatment to monitor the success of these kinds of treatment.
Thank you, Doc.
Okay, Alex. I saw Kenneth kanina, parang nag-raise ng hand si Kenneth. Kenneth, did you raise your hand or napindot lang? Yes, go ahead, Kenneth. You can unmute your mic. You can unmute your mic. Go ahead, Kenneth.
Sorry,
Yeah.
Doc. So my question, Doc, is I don't know if I remember correctly. There's an article that says that an AIDS patient got healed through stem cell therapy, or I don't, or it's not stem cell. I forgot.
Again, please repeat that.
Um,
There's a
oh, um, AIDS patient who got he healed through stem cell therapy, or
AIDS
or is
patient,
it,
HIV positive.
yes, go.
Oo. Um, I haven't uh, come across that article, no? Yeah, maybe we can look at that, no? Kung ano yung their outcome, no? Out after bone marrow transplant or stem cell transplant.
So stem cell can also be used or possible future use for HIV.
Theoretically possible, no? Kasi you're able to prime the cells, and your cells will be able to develop, like for example, helper cells, no? That will fight against infection, no? As opposed dun sa mga HIV infected where you have more of the suppressor cells than the helper cells.
Okay, though.
So anything else that you'd like to add? So as I mentioned earlier, no, so uh, we will be expecting the report of Yuseng, no? I mean, Yung Liu next week, no? She will be reporting about immunotherapy, which is also related to stem cell therapy. All right. But for now, we just listen to the report of Yuseng, no, on stem cell therapy. But next week, we will be hearing at least 3 reports, no, reporters. That's Alex and Kenneth about the development of vaccines. But we will start muna with Yung Liu to discuss about immunotherapy. All right. So because I understand we still have enough time, so I actually prepared a few more slides, but this is not about immunology or blood banking for that matter. What I prepared, so let me just pull that slide now so I can share with everyone. This is something about your thesis, no? So maybe this would help you later on prepare for your thesis, all right? So again, this is not something about the course, but I just put this, no, because we still have time, no? So to maximize our time, I thought of sharing this with you for the benefit of your thesis writing. So this is all about scientific technical manuscript writing, and you can also relate to this when you later on prepare your thesis proposal. All right. So before that, I'd like to quote, no, from this author. This is George Gopet, no, and let me quote. Um, if the reader is to grasp what the writer means, the writer must understand what the reader needs. No? So this was published in American Scientist in 1990, and this is the title of the work, no? The Science of Scientific Writing. So in other words, what the authors are saying is that we need to make sure that the the way we write our paper, no, let's say a thesis proposal, no, should be clear enough so that the reader won't be asking any questions, no, anymore. So let me just mention that in scientific technical writing, no, there are 3 different kinds of uh, papers that you can write. No? And one of these is your proposals. No? This is a requirement for graduation, of course. Your thesis proposals, or for those who are pursuing doctorate, you need to also have your dissertation in the form of a proposal as well. But aside from proposals, you also have your scientific papers, no, like a publication in a peer-reviewed journal, no, which is also important. And finally, if you want to, let's say, apply for a grant, no, in a, let's say you want to apply for a Bill Gates Foundation na grant, no, okay, so you need to also write, no, to have some training on how to write a paper, no. Yeah, so these are the different scientific technical writing, which is essential, no, as part of research. So again, these are your proposals, your papers, and patents. So I included this photo, no, just to make a point. So when you write your thesis proposal, I want you to remember this picture, no, the fish. So you might ask, why the fish? Well, simply because you have the head, no, you have the head, you have the body, and then you have the tail. No? Similarly, when you write your proposal, I want you to remember, first of all, the head. So you can actually chop into three parts, no? The head, the body, and the tail. So when you prepare your thesis later on, no, you will think of this first. No? You will think of what would be a good title for my proposal, and I need to prepare at least 250 words of abstract, and what are the keywords related to my thesis or my proposal. No? So ito yung head part ng inyong thesis proposal. But again, when you are writing your thesis proposal, please don't forget the so-called finer research. No? Finer. F meaning something that is feasible. I should be interesting. N should be novel. E, ethical. And finally, the R should be relevant. Okay? So it's feasible, interesting, and novel, ethical, and relevant. So once it fulfills all of these criteria, then you can proceed, no, with your idea, with your research. So after this head, no, yung meron na kayong title, your abstract, and your keywords, the next part that you need to do is yung chapter 1 and then of course chapter 2. No? Yung results discussion, wala pa yan. But if you're doing a research paper, no, yung finished paper na, then you have the results and discussion. So basically, you need to have a very comprehensive introduction. You need to orient the reader, what is the problem, what is the gaps, the knowledge gaps, and how do you intend to answer your research question. And in the methods, you will highlight what would be your approach, no, your strategy to address, to answer your research problem. Meaning to say, you have objectives. So for every objective, you need to have a specific methodology to answer that question. Now, in general, you would need at least mga 3 to 5 objectives in one research proposal. Likewise, if you are making a project, that would be the average number of objectives in a given paper. Again, remember this. Actually, you can make an acrostics or acronym for this part. No, it's called the IMRAD method: the introduction, the methods, results, and discussion. If you're making a paper, na, but for proposal, well, you need to invest time in making your introduction. That will be chapter 1, and of course your methodology. No? Yeah. And then of course after this, you need to highlight also what are the other relevant related literature. So that will be your chapter 2. So it is a comprehensive, extensive literature review. What are the current findings? What are the gaps? And then you will insert, no, and discuss what are your rationale in doing these kinds of research. Now, suppose you've done already with your introduction, your methods, results, and discussion. The last part of a scientific technical paper would be the conclusions, acknowledgement, of course, especially for those who have also contributed in the making of that paper, but are not qualified to be an author. And then, of course, your list of references. So I'd like to end with this 6S in scientific writing. All right, the 6S in scientific writing, which you can also adapt when you prepare your respective research proposal. So the first S is it should be structured. No, you need to write under the guidance of an outline. So I already discussed a bit about the outline. No, so you have the title, you have your keywords. No, if you're preparing the abstract, the IMRAD, the introduction materials, no, these things. And then, of course, you need to include your RRL, no, related literature, no, and so on and so forth. So the results will come in later, and the discussion, and then, of course, the acknowledgement and the references. All right? So it has to be structured. Know where the logic starts from and where it is going, no? Its structure. The second S is it should be sequential. No? Take the reader by hand through the sequence of thoughts without any leaps or missing links in the development of ideas. So stage by stage, step by step. So it should be leading towards your problem. So you introduce first the concept, and then later on you shift gears and start to introduce what is the gap, what is your rationale, what is your hypothesis, and what is its significance. The third S now is simple. No? Imagine trying to explain the concept to a layperson. No? So if the layperson can understand what you wrote, then it means your writing is simple. The more simple, the better. No? Yeah. The fourth S is it should be short, not necessarily long. No? It doesn't mean that your thesis is long. It's more, it's better. No, it doesn't mean that way. No, so use short sentences, no, containing only one idea in each. No, don't make the sentence too long. No, you can actually split, no, complex sentences so that the message is clearer. Right. So that's the fourth S in writing a scientific paper, including a proposal. And then the fourth one, or the fifth one, is it should be strong. No, you need to highlight what is novel of your work. No, you can actually use active voice instead of using a passive voice. No, so you can say na we would like the authors will conduct. No, the authors will conduct, because it's a proposal pa lang, no, will conduct, or pag done na, then we conducted. So that's the active voice. On the other hand, yung passive voice would state like the study was conducted. So better to use the active voice, which is better than the passive one. And then finally, it should be specific. No? Say clearly and exactly what you want to say. Right? So that would help you prepare your thesis proposal later on, no? When you about to finish the coursework that you. Okay, so that's my inputs to everyone. Okay, all right. Now stop sharing. Okay, so I'd like to ask, so how many of you are now working with your thesis? Do you have a topic na ba that you're working on? At least, Peter, I heard meron ka na, no, Peter? TB? Tama?
Well, wala pa, Doc.
Oo, oo. Oo nga. Yes, and I think Kenneth then also is preparing one. Yeah. One of your classmates